Okay, let's tackle this last topic in chemistry of life, and that is nucleic acids and how they relate to ATP. So, when we're talking nucleic acids, we are talking ribonucleic acid and deoxyribonucleic acid. In other words, RNA and DNA. Like I said, I knew you knew what I was talking about. You just may not have remembered the word nucleic acid. So nucleic acids are made out of individual little building blocks stacked on top of each other, known as nucleotides. And a nucleotide has three parts to it. Here in green, this part is a sugar. It's a pentose sugar, and then it is going to be attached to the next pentose sugar with a phosphate group. And you can see sugar, phosphate, sugar, phosphate, sugar, phosphate all along what's called the backbone. Okay, so it's the blue part here and the swirly part here is laid out fat. So think of this like the side rings of a ladder. That's the sugar phosphate backbone. And extending from the backbone, you are going to have what we call nitrogenous bases. And there are so the sugar, sorry, before I get to the nitrogenous bases, the sugars can either be ribose or deoxyribose. So this is ribose. It is what is found in RNA. So ribonucleic acid, the ribose stands for ribose, whereas in DNA that stands for deoxyribose. So here is the difference. In ribose, we have two hydroxyl groups here, as well as one up there. Whereas in deoxyribose, notice this one is just a hydrogen, where over here it was a hydroxyl. So that is the difference between the sugar groups in RNA versus DNA. Okay, so if this was DNA, these would be blue instead of green, and if this was RNA, you can make these pink instead of green in the backbone. Now, the nitrogenous bases, there are 5 of them. And if you look, you don't have to know the words purines and pyrimidines, but notice that 3 of them are single rings, as shown here, 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 and here, whereas 2 of them are double rings attached to each other, shown here and here. So in order to get equal spacing, if we have a ladder with things attaching across, in order to get the equal spacing, it would require 3 across, a double ring with a single ring to be attached. And this was the big mystery that had to occur. So let me show the differences between DNA versus RNA before I talk about that. So in DNA, okay, so I said there's 5 different nitrogenous bases, but each nucleic acid only uses 4 of them. In DNA, cytosine will bond with guanine. So this single one will bond with this double one, and adenine will bond with thymine. This double one will bond with that single one. So it'll be 3 across here, and it'll be 3 across here. The sugar is deoxyribose, and it's a double helix in that 2 strands will attach and twist. In contrast, RNA, cytosine still attaches to guanine, but now adenine attaches to uracil instead of thymine. Okay. So uracil is very similar to thymine, except that this part is a little bit different. Okay. And the sugar is different. It's a ribose sugar instead of deoxyribose, and it's only a single strand instead of a double strand. So 3 big differences. Uracil instead of thymine, ribose instead of deoxyribose, and single strand instead of a double helix. All right, so we did not understand what DNA looked like until the 60s, and here is how it happened. You know, there were these two young people here in the United States, James Watson and Francis Crick, and they were the East Coast of the United States. And in the summer, like their parents said, like, you gotta go do something. So they were sent to England in the summer to go find something to do. Like, you can't come loaf around all summer. So they went to the lab of Maurice Wilkins. And he owned a lab, and his idea was to study DNA doing X-ray crystallography, but he was off doing whatever. It was hot in London in the summer, so he was off probably on vacation. But there was a lady in the lab, worked in the lab, and her name was Rosalind Franklin, and she was the one doing the work. She was making images of DNA with X-ray crystallography. So she was doing X-rays of DNA, and she could see the shape, and she could see between the backbone, it was an equal shape. Okay. Before this with DNA, people were playing around and they were trying to move sugars in and out, and they were trying to move all kinds of things in and out, and nobody was able to figure out the shape of DNA. So Watson and Crick, they went up and basically they just kind of chatted it up with Rosalind Franklin, and she showed them her work, and she showed them, look, this is what has to happen. Like, we have a sugar phosphate backbone, and so the nucleotides have to be in the middle, and it has to be equidistant all the way across, and so you're going to have a single with the double. Okay. So Watson and Crick, they went to King's College, they looked at her images, and then they went home and they played with essentially Tinker Toys. And they created this double helix model with the sugar phosphate backbone. So they made a visual model of what her x-rays showed. And then they wrote an article in Nature magazine in 1953 called Molecular Structures of Nucleic Acid, a structure for deoxyribonucleic acid. This is the entire length of the article. Here is their drawing, signed by Watson and Crick. This is to prove you don't need 10 and 20 page essays to have important work. This shows in science, we have conservation of energy. We tend to get to the freaking point quickly. Okay. So what happened with this? This was huge. This was Nobel Prize material. So I ask you, who gets the Nobel Prize? Well, in 1962, the Nobel Prize in Physiology and Medicine was given to Watson and Crick for this article on this page, given to James Watson, author, given to Francis Crick, author, given to Maurice Wilkins, whose lab Rosalind Franklin was using to do the pictures that Watson and Crick used to base their model on. So you ask, well, what about Rosalind Franklin? Nope. Misogyny was well alive. She got bupkis. Oh, and by the way, did you know the Nobel Prize comes with like $1 million? She got absolutely nothing, and she got no acknowledgement for almost 50 years, 40 to 50 years. She got no acknowledgement for her contribution. She's the one that figured it out. Who do we give credit to? Watson and Crick. Just saying, ladies. Don't be so easy to give off the work that you do. All right, so what does this have to do with ATP? Well, ATP stands for adenosine triphosphate, okay? And Whoops. Adenosine is adenine with a ribose. Sound familiar? So adenine with a ribose, and then we have phosphates, 3 phosphates. These are high energy phosphate bonds. So when we have ATP, which is our source of energy, we're going to have hydrolysis. What's going to happen? That we're going to end up with ATP, an inorganic phosphate. That's what PI means. I means it's inorganic. Why do we say it's inorganic? Because it's not containing what? This is breaking off right here. What is it not containing? And it's going to give us an energy. So here, these are our ATPs. This is what we're going to come back and learn all about when we talk about energy in very soon, next week sometime. Okay. So if you're feeling lost and confused about this chemistry stuff, go back, watch the videos, watch some of the supplementary videos, go to the tutoring center, come see me. Remember, remember, students will become what they believe, so you need to believe in yourself. If you believe you cannot do something, you will always be incapable of doing it. But if you believe you can, then you acquire the ability to do it, even if you don't have the ability in the beginning. Okay. And lastly, here is a small concept map putting together everything about the chemistry of life, just in case you want to see it. And with that, we are done with this whole second lecture series, and I will see you next week. Thank you for all your hard work.